Hi everyone, my name is Kristen Russell and I'll be one of your on-camera teachers tonight. We are super excited to have all of you guys with us, so make sure you're saying hi in the chat. We do have a pretty awesome crew tonight. We have Deborah, we have Dee, we have Justine, we have Carrie, we have Mary, and we have Paul. So definitely chat us up, let us know what you guys are thinking, ask us any questions you have because it's going to be a fun and interactive lesson tonight. So let's go ahead and take a look at the plan. We're going to do our quick welcome and intro now, so make sure you're introducing yourselves to us. We're going to do then a quick overview of the MCAT, talk a little bit about why it's so important. Then I'll be handing things over to Justine. She'll be having an amazing time with you guys as she talks about some content and a little bit of strategy along the way. Then you'll come back to me after the lesson and we'll talk a little bit more about what you should do from here on out. Of course, learning is part of it, but having a good plan is critical for success. So we'll talk about what kind of things you should have in your plan as you're moving along, and we'll share with you the types of resources that we have that might help you along the way. So let's talk about why the MCAT is so darn important. So I want you guys to share with us in the chat where you go to school. I always like to see where people go to school. Vanderbilt is my alma mater. If I'm not mistaken, I believe Justine went to Pitt. I think I'm correct, Justine. So we have tons of our own schools that we go to. We have our own majors. I'm a biomedical engineer, undergrad and master's. Justine has a PhD. So, we all have our different areas of expertise. And when you look on a resume, it's really hard to compare all those people, right? Just like you guys with all of your different awesome schools and all of your different awesome majors, it's very difficult to compare all of those things. GPA doesn't really do it justice when you're not looking at the same classes. So that's where the MCAT comes in. Now, we certainly could think of this in kind of a negative light that, this is another standardized test, and why do I have to do this? Can't they just use all the other things I've done? Unfortunately, no is the answer to that, but you could also say, fortunately, it does matter. Because let me just ask you this, how many of you guys have a 4.0 GPA? Some of you probably do. I'm willing to bet that some of you guys do, but I'm also willing to bet that a lot of you don't. In 3 months or so, it's essentially impossible to have a significant improvement to your GPA. I'm sure we would all have done something if we could have a drastic improvement that quickly. So that's really hard to do something about, especially if you're a junior or senior, or if you've already graduated. Very difficult, of course, to improve your GPA. But in 3 months, some of you maybe even less than that, you could have a significant improvement to your MCAT score, which means that you're going to look even better on your application. Admissions committees are going to be looking at you and seeing this wonderful presentation of your knowledge and skills. So if we think about it in a positive light and think of it as an opportunity to shine and make ourselves stand out in comparison to others, I think that's the right attitude. If you go into it really thinking, oh, this is going to be terrible, I'm terrible at standardized tests, I have test anxiety, there's so much material, how could they ever expect me to know all of this? I mean, we could probably come up with some pretty strong complaints as to why we don't like the MCAT. But if we go into it that way, it's going to be miserable the whole time. So think of it as your other opportunity to shine, and know that you can make a huge, huge improvement, and know that admissions committees really do care about the MCAT. When polled, medical school admissions officers that have been surveyed, like half of them, as you can see here, say that MCAT score is the #1 most important thing to them when reviewing an application initially. Now, of course, GPA is also pretty important. That's second on the list. Then we have relevant experience, interview, letters of rec, personal statement, all these things. So the question here is, what is the most important factor in the admissions process? So, all of these things, of course, are important. But I think we all understand as well that upon first glance on your primary application, the things that are really going to make you stand out from the crowd, MCAT score, undergrad GPA, maybe are the things that aren't going to make you stand out, but they're things that you have to have for them to care about the other things that do make you so unique. So all of those things, where you go to school, what your major is, if you do research, if you're an athlete, if you're in some amazing extracurricular activity, all of the things that make you you, we want them to see those. but you have to have an MCAT score and GPA that is good enough that they're willing to look at all those other great things about you. So we want to make sure you have that good MCAT score. So we're going to talk tonight about how to help you get there. If any of you are stressed because you've taken a practice test, tell me in the chat if any of you guys have taken a practice test before. And if you have, how did it go? I'm curious like what your initial thoughts were. I know that when I took my very first practice test, I felt pretty awful after taking it. I felt like I was pretty tired. I felt like I had done absolutely terribly. And I mean, I did. I scored in the 15th percentile, 15th percentile. Just atrocious. So that is a hard feeling, right? Especially if you feel like, okay, well, you know, I've done well in my classes. I thought I could handle this. I'm pretty good at test taking, whatever the case may be. This test is just so different. So it's easy to feel very overwhelmed by a first practice test. But know that that is the case for most students. Most of my students start off in the 480s, some of them in the 490s, some in the 470s, but a lot in the 480s. But I see a lot of those students scoring really, really wonderfully in the end, like scores 90th percentile and higher. I went from scoring a 488 to over 90th percentile, so over a 513, the equivalent when I took my test. So even if you're feeling absolutely terrible, which some of you guys are like the whole discouraged, not as great as I was hoping, that is normal, but don't let it prevent you from moving forward and studying, because with the time, with the right things, like the things that you need to do to increase your score, you will see a drastic improvement. So stick with it. Sometimes it takes a few tests to get under your belt. It takes a lot of hours. I'll just go ahead and tell you 350 hours is what we usually recommend as the number to try and hit in terms of overall study time. That's a significant amount of time. But if you compare that to other things that have been really important to you, maybe learning to play an instrument, learning to play a sport, getting your major finished, all of those things took way more time than studying for the MCAT. So it's definitely doable. You will see some life changes as you go through studying for this with all the other things I'm sure you guys have going on. But I know that you have it in you, so try and soak up as much as you can tonight. And I'm sure you guys have learned some good stuff for the rest of this week since pre-prep week. So let's get you into Justine's lesson as soon as possible here. I just want to talk to you about a couple more things. So on the MCAT, if you guys aren't super familiar with the layout of the test yet, there are 4 sections. 3 of the sections are science sections. Each of the science sections is 95 minutes long. There are 10 passages with associated questions plus 15 discrete questions. Those are the standalone, no passage associated. But that's for a total of 59 questions on each of the science sections. Then we have, which is probably one of my favorite sections, critical analysis and reasoning skills, which we refer to as CARS. That's 90 minutes long and has 9 passages with associated questions, specifically 53 in total. A lot of students initially start off pretty similar scoring across the board because if you don't know a ton, it's hard to see a big difference in the scores. But some of you may have seen a difference. Just know that a lot of these sections are going to feel different when you first start off, but you're going to use very similar strategies and approaches across all the sections. So I think practice on any of them definitely has some overlap, even when it comes to CARS. It feels so different from the sciences, but you're going to be reading and approaching questions in a very similar way. Tonight we'll be talking just about sciences, but we can talk a little bit more about CARS and kind of how to approach that when we come back together. So scoring is something that I'm sure you guys have heard a little bit about before. So I will just talk to you guys, kind of briefly about the scoring and maybe where you want to end up. I would say if I didn't know anything else about you, like nothing else about you, I would say you would want to be scoring somewhere at a 510, 511. And the reason I say that is because that is the average matriculant score, person accepted to medical school. Well, there could be other things certainly that might make this a little bit different, right? Okay, if your GPA is a little on the low side, then you would want to really kind of overshoot when it comes to MCAT score. If you are looking at a super competitive program, yeah, then you're probably going to want a little higher. Or, maybe you have a very specific school that you want to get into, so you want to make sure you're above that average. So if you would like to, share with us in the chat what your target score is. Now, I see some of you guys also wanting to know a little bit about like when you should take it, and that really to me comes into the discussion of score quite a lot, because I always tell students it's important to balance two things: taking the test early with a high score. Ideal scenario is that you have your score back before the MCAS even opens for you to submit, which happens on June 1st. So having your score back before then would be ideal. I realize that's not going to happen for every single student. So at that point, what you want to do is ensure that you are taking it as early as possible and maximizing that score at the same time. Generally, what I say is that May, June is kind of the, okay, you're not late, but you're definitely on the border. And then July, August, and September, I generally consider to be pretty late taking the MCAT if you're going to be applying that same cycle. But I do have students all the time who take the test in August or September, but they're taking it and applying the following cycle. So when it comes to early, early is better because of rolling admissions, but getting that good score is also super important. Now, just so you guys are aware, the average on the test is 500. That's average of everyone taking it. So average matriculants, 510. Average applicants, a little bit lower than that, of course. Now, if you're interested in some of the higher tier scores, a 90th percentile is around a 513, 95th percentile is around a 517. The highest I've ever had one of my students score is a 523, which literally says 100th percentile. So, I don't know about you guys, but I think most of you would probably be pretty happy with that. So it certainly can be done. And I just want to point out that my student who scored a 523, she started off in the 490s. So it's not like she started super high. She started below average in terms of average test score on test day. So I know it can be done. So let's talk for a moment about the distribution of science content so you can see which are your faves and how much they're tested. So the number one most tested science on the MCAT is biology, which I don't think is really too surprising to anyone. Number two most tested science on the MCAT might surprise you. It is psychology. But you can see that the kind of lighter red chunk in the psych-soc section is huge, which is why it's such a big part. Number three most tested science is biochemistry. Biochem is one that I find that some students have not had before. It is something that if you can fit in before you're going to take the MCAT, that would be highly beneficial to you, and I certainly recommend that. If you're not going to be able to fit it in, you will want to figure out how you're going to be able to independently study that topic and still learn it to a pretty high level. It is tough to teach yourself, but not impossible. It is a big part of the test, though, so you'll want to make sure you plan for that. So let's talk for a moment now, not about what science content's tested, but what the skills are that are tested. Since you'll be focusing on science with Justine during the lesson, we'll talk about the 4 primary skills that the AAMC is going to test you on, and then, of course, something that we've thrown in, just being an MCAT expert. It's not always about just knowing the content. It's about knowing how they're going to test you on the material, the types of traps they're going to have for you. If you know how the test maker thinks, you can beat their test. All right, now the 4 skills that the AAMC officially says they test on the science sections. We have skill 1, which is science knowledge. This is just your ability to recall science information. Skill 2, this is your reasoning, your problem solving, just really where they're testing your critical thinking skills. Usually more steps involved in these problems, sometimes looking at a passage and bringing in information from there, plus outside information. This is sometimes where the math comes in as well. Skill 3, here we've got research design. So this is kind of cool because it doesn't always require you to have to know a ton of specific content related to a topic, but you'll be looking at experiments on topics and kind of analyzing how the experiment is set up, figuring out if there are problems, understanding variables and relationships, all that good stuff. So I really like a good skill 3 question. But my favorite question type is skill 4. Skill 4 is data and statistical analysis. So this is interpreting figures. This is looking at tables and coming up with what you think the results mean. This is basic stats. So nothing too crazy, but mean, median, mode, that kind of stuff. So those 4 skills will be what the questions are testing you on, of course, over a wide variety of different topics. The MCAT is a mile wide, but only an inch deep. So you certainly want to make sure you have time to go over that very broad spectrum of different topics during your content review. So without further ado, I believe it is time for me to hand you over to Justine. You're going to be in very good hands. You're going to be learning about one of our MCAT channel topics tonight. Justine is a teacher on MCAT channel and she's fantastic. So I know you guys are going to have a blast. So I'm going to hop off and she's going to hop on and I will see you guys back here a little bit later.
Hey everyone, I am super excited to be going through some science with all of you tonight. We are specifically going to be focusing on molecular genetics, DNA, and transcription. So there's a few things we're going to cover in our about hour session here. We're going to talk about the steps involved in DNA replication. We're going to look at DNA repair since our genomes are constantly being bombarded with mutagens from the environment. So it is essential to have a repair mechanism to protect against mutation. And we're also going to look at transcription and gene regulation, which the MCAT tests a lot in prokaryotes. So we'll look at the 2 main operons that are most likely to appear on test day. So before we get into DNA replication, I do want to talk a little bit about DNA itself. So what can you all tell me about just our own genomes? What can we say about our own DNA? Because that will inform a bit about how it is replicated. I'm just going to draw out, of course, the DNA is going to be much longer than this, but if we think about our 2 strands, the DNA itself has polarity, and I'm seeing negative charge, that's a phosphate backbone, and we get DNA from both of our parents. We'll have homologous chromosomes, absolutely, and absolutely it is linear. And Dominica, I love that you're putting in the polarity. The polarity will really become important when we look at how the DNA is specifically replicated. So our 2 strands are actually anti-parallel. So one strand will run 5' to 3'. The other one will run 3' to 5'. So we call this an anti-parallel arrangement of our DNA strands. Now we also need to consider the nucleotides themselves. So of course, A and T pair, G and C pair, but we actually have different numbers of hydrogen bonds between those 2. So how many hydrogen bonds do we have between AT base pairs? This will be important because it actually, MCAT loves to test about the melting point of DNA strands as it relates to the number of hydrogen bonds between our base pairs. Nice, and now we got 2 hydrogen bonds, absolutely. And then between GC base pairs, we have 3 hydrogen bonds, which means that if we have a high GC content, that will mean that the melting point of those DNA strands will be higher. So put here higher GC content equals higher melting point. The other thing you want to know are your purines and your pyrimidines. And I always hate just blunt memorization. Even though I am a biologist, I still hate memorizing stuff. So the mnemonic I like to use, pure as gold. So A and G are our purines. And if we think about joys of marriage, 2 gold rings. So pure as gold, 2 gold rings. And the other one I love, because personally I love pie, is cut the pie. Cut the pie. Notice that the U there is uracil. That we'll find specifically in RNA. Yeah, so Sarah, you got those two as well. Nice. So on with our review here, let's dig into the mechanism of DNA replication. And the first thing I want to talk about is the start place of DNA replication. Now in our genomes, we have thousands and thousands of these. In bacterial genomes, they only have like 1 or 2. We have a lot more genes, we have a lot more DNA, so we need a lot more start places. Now, where on the DNA do we actually start DNA replication? Where do we actually start DNA replication? I'll tell you that it will be a location that is rich in A and T nucleotides, since those are easier to separate. But where do we call, oh, nice, Dominica Ada, we got the ORI, the origin of replication. Now I see a few other answer choices coming in here. Start codons, that's specifically for translation. And I know a lot of my students will often say like Tata box or promoter. That's specifically for transcription. So DNA replication will start at the ORI, which is just short for origin of replication. And this is AT rich, because remember, AT base pairs only have 2 hydrogen bonds, so it's a lot easier to break those hydrogen bonds to start replication. Now, we are expected to know all of the key players in DNA replication. So just go ahead and just list some of the enzymes that are involved in this process. And then tell me some of the enzymes we need to care about here. There are quite a few. We'll be going through them one by one. Let's just get a list going to get started. Yeah, Kiana, helicase, allyl DNA polymerase, absolutely. Ligase. See a lot of polymerases coming through. Absolutely. So let's start. Oh yeah, Kiana, Okazaki fragments. We'll talk about those. Oh, nice, Dominica. Single strand binding protein. Primase, absolutely. And if you're saying polymerase, Melissa, polymerase 2 is what we usually associate with transcription. It's RNA polymerase 2 that transcribes all of those messenger RNAs, which is really the only polymerase that like comes up specifically. So let's start with helicase. So we need DNA helicase because this is what unzips the genes. So this is what unwinds the strands. It uses ATP to do this because it takes energy to break the hydrogen bonds between our strands. We also need to consider the effect of that unwinding. I don't know, this will be something that'll show my age, but when I was little, you know, back in the day before like cell phones, our phones were actually anchored to the wall with cords. I know, crazy to think about, and they were all coiled. And as a child, I loved to like try to unwind them to make a perfectly nice straight region of cord. But the problem I always ran into, this is why I got yelled at by my parents, is there were always regions around that nice straight region that just got bunched up and ugly, and I did ruin a few phone cords that way. The same thing happens to DNA under the action of topoisomerase with the helicase. And I'm glad that your professor, we're on the same page. So if we have a lot of strain on the DNA, it can actually cause shearing of the DNA, which would obviously be bad for our cell. So we have evolved topoisomerases. It's also sometimes referred to as DNA gyrases that will actually essentially allow it to spin around and uncoil and then re-ligate. And this will remove tension from the strand. So we'll just put minus tension for what that enzyme does. And at this point, we're getting ready to start. Here's the problem. Can DNA polymerase just hop on and start making DNA? Can DNA polymerase do that? Just hop on, you're like, all right, let's go. No. We see it's not capable of de novo synthesis. So it needs a primer, absolutely. It can only extend from an existing 3' OH. That's what primase does. Primase lays down an RNA primer so that we have that tongue twister there, a free 3' hydroxyl group from which to extend. So this gives us the primer to extend from. And at this point, DNA polymerase is like, I see we got all the stuff I need to get started. I'm going to hop on and I'm going to start making some DNA. And this is where we get into the idea of a leading strand and the lagging strand. The replication fork will be moving in its own direction. Of course, it will be spreading out in both directions. We're just looking at one of those right here. And here our replication fork is moving this direction. But DNA polymerase can only synthesize DNA in one direction. It can only go 5' to 3'. So we have one strand that is progressing with the replication fork. And that guy is our leading strand. He is made in one continuous piece. Put here, one piece. The other strand, however, has DNA polymerase moving away from the replication fork. Remember, it still has to synthesize DNA 5' to 3'. But in this case, it cannot move with the replication fork. So what do our cells do to get around this? What do our cells do to get around this? We still have to synthesize both strands. We can't just be like, we're just going to do half of it and call it a day. We do need to make both of these. But one of them, we need to do a little extra. You got it, Hannah. It's Okazaki fragments. So we continuously, essentially, build going away from the replication fork. So we go 5' to 3', and then DNA polymerase will have to hop back. Well, of course, we'll have that primer. It hops backwards, synthesizes again away, going 5' to 3'. So instead of going in one continuous piece, we are going and making Okazaki fragments. They're a couple thousand base pairs long. But they're all going 5' to 3'. And here's a little thing that I found that helps describe this a little bit better. Let me just type this out. So if we look at this, notice that this sentence does seem like it's written in somewhat broken English, or I guess said by Yoda if you're a Star Wars fan. But notice that each word is still written from left to right as we do in conventional English. But what happens is in order to write this sentence, we would type out this. We would jump backwards, is, jump backwards, how, going backwards, Okazaki fragments work. So we're still building 5' to 3'. We're just consistently jumping backwards and continuing to synthesize away. So that's how our Okazaki fragments work. And after we're done, we're going to be left with a bunch of, fairly short strands. What enzyme do we require to join all of our Okazaki fragments together? I will note that DNA polymerase has its own exonuclease function, so it is able to, in conjunction with other enzymes, remove the RNA primer and replace it with DNA. So even in the lagging strand, you won't see any RNA bases in the final product, but we need DNA ligase, absolutely, Sari, Alex, Diamond, perfect, to join those Okazaki fragments together. So this would be specifically in our lagging strand. Please join the fragments. Now let's think like a test writer here. We have one strand that's made in one continuous piece. We have one strand that's made in kind of a piecemeal process. Which strand would you expect to be more prone to error? This would be a really good kind of a skill 2 question, because we're taking what we know about DNA synthesis, DNA replication, and we're going one step further with a little bit of critical thinking. So absolutely, I'm seeing answers coming in. Diamond, Domenica, you got it. That is the lagging strand. I got it, Erin. Because the more people you have, the more think about it this way, the more cooks that you have in the kitchen, the more likely it is someone's going to bump into somebody else. So lagging strand is more prone to error. The other kind of accessory proteins we have in here, of course, the RNA primer, not necessarily an accessory protein, but still an important part of our replication process. The other one was mentioned at the very beginning as single-strand binding protein. As helicase is unwinding, DNA strands don't like to be single. They like to be paired up. So single-stranded binding protein is there to stabilize the single strand so our two strands don't re-anneal before they get replicated. So that is DNA replication. Now, there are a few specific adaptations that we have in eukaryotic cells. And we've already talked about one of these, the fact that since we have millions and millions of base pairs in our genomes, we have multiple origins of replication. This allows us to just get more work done much more quickly, because otherwise DNA synthesis would take days, weeks, and in our cells we can replicate our entire millions of base pairs in just a matter of hours. The last issue that we have is that at the very ends of our DNA, at the very ends, because our genomes are linear, there is some bit of DNA that we lose each replication cycle, and that's because the RNA primer that we put down cannot be replaced. Essentially, it's just cleaved off of it. So we lose a little bit of DNA each time we replicate. So our chromosomes are getting shorter and shorter and shorter each time we replicate. And that's one of the things that contributes to cell senescence. We lose the ability to divide. It's part of our aging process, unfortunately. We do have an ability to at least overcome this to an extent, and that is through telomeres. These are repetitive regions at the ends of our DNA that essentially don't encode anything. So when we replicate, we lose a little bit from the telomeres. That's fine. That's what they're there for. But eventually, when we start getting into coding regions, our cell senses that it's losing some of its genetic material, and then we'll stop dividing. There is an enzyme, however, that we can use to extend the telomeres. And what types of cells would you expect to see overexpression of this enzyme? The enzyme is telomerase. It's what allows us to extend our telomeres, increasing the potential lifespan of our cell. It's in cancer, absolutely. Yeah, this is found in cancer cells. But if we are able to find a way to unlock this in all of our genes, all of our genomes, it could be a potential fountain of youth. Now, I also want to talk a little bit about the DNA replication process itself, because I know, all of us being, youngins in the field of biology, a lot of the knowledge that we learn, we really take for granted. We've always said, yeah, DNA replication is definitely semi-conservative, but back in the 50s, we did not actually know this. There was some indication that semi-conservative was the most likely process after Watson and Crick proposed their model of the DNA helix. It was the one that made the most sense, where we replicate each strand. But there were some other theories of either just conservative, where the parent strands stay together, or one that kind of always intrigued me where we came up with this one, but dispersive, where essentially old DNA and new DNA were randomly interspersed with each other. So the Watson-Crick did give us some support for semi-conservative, but that model wasn't really widely accepted until we experimentally proved it. And that's what Meselson and Stahl did. So this is actually one of a pretty cool experiment. You can certainly see it tested on the MCAT, especially in passages. So let's take a look at how that works. So while I start to draw some stuff out here, tell me what you know about centrifugation. We take stuff and we centrifuge it. Tell me what we know about centrifugation. To separate organelles based on weight, breaks up blood cells. Absolutely, so one way that we can separate plasma from red blood cells from leukocytes. Absolutely, heavier at the bottom. We separate by density. It's not technically weight. We separate by density. So the way that this experiment worked is they had some bacterium, and they grew the bacterium in heavy nitrogen. So that would be nitrogen 15. It's a heavy isotope of nitrogen. So at the very start of the experiment, they saw just one band, which would correspond to heavy nitrogen. Now, as the DNA was replicated, and that happened, let's say our cells divide roughly every 30 minutes. It depends on the bacteria, let's say that, we're dealing with just general E. coli in normal conditions. We replicate roughly every 30 minutes. So what they observed was something that looked like this. At 30 minutes, they had just one band. At 60 minutes, they then had two bands. And as DNA was replicated, that middle band got smaller and that top band got larger. And this is what helped us to prove that it was semi-conservative. Because what happened is we had essentially that lower band, which corresponded to our heavy isotope, that N15. And then we had our normal isotope, which would be just normal nitrogen 14. So because it's semi-conservative, after our first replication, we saw this, where each of the two strands was composed of half of the heavy original parent strand and then half of the newly synthesized light strand. And this is how we proved semi-conservative. And as our replication continued, we saw just more and more of the normal isotope incorporated. So that is how we originally discovered semi-conservative. It's our first piece of experimental evidence. Now, what would this look like for conservative? Because this is another type of MCAT reasoning question that we could see on the test. They like to look at, well, given that we know information and we know what conservative is, can we apply it to a specific instance? This would be another type of skill 2 question. So what would conservative replication look like in an experiment like this? I'm going to do a little color coding here. So say it was conservative. We're going to draw our bands here in blue. So let's think about it. In conservative replication, our parent strand stays together, and the newly synthesized strand is made of completely new DNA. So if it was completely conservative, half and half, there'd be two different densities. Exactly, Levi, Aaron, you got it. 50-50. So we would see a heavy strand and we would see a light strand. Absolutely wonderful work. So I love the way that you guys are thinking. This is exactly the type of critical reasoning the MCAT will expect us to do. So this is how you want to think about science as you're studying. Know the experiments, at least be able to reason through them, understand how they work. And if there were different models that the experiment tested, think about what the results would be for the models that were wrong. If it were dispersive, essentially what you would see is like a smear of sizes. You wouldn't get distinct bands. So challenge yourself. And speaking of challenging yourselves, I'm going to challenge you with a question. So I'm going to have Mary open up a poll for you to vote in for on this question. And I'm going to give you 30 seconds to a minute to think about this and vote in. So I'll go ahead and be quiet and let you do your work. Just go ahead and vote in when you have an answer. All right, I gotta say, I am absolutely loving where we are on this poll. We got over 90% of us going with answer choice C, which is absolutely spot on correct. And I will say if you missed this one, don't feel bad because really, anytime you make a mistake, it's really a learning opportunity. I know Kristen was saying she started out, below the 50th percentile. I can tell you I started in like the I think it's like the 19th percentile for mine. It was pretty terrible. And in order to get that low, you got to miss a lot of questions. And in order to get a high score, you got to miss a lot of questions too. The difference, though, is that when you miss questions, you want to use it as a learning opportunity. Figure out why you made that mistake. What specific behavior caused you to miss a question? Was it just a lack of content knowledge? You can fix that. Study that area. Did you not read the question stem carefully? In that case, you need to focus on paying attention. Did you rush it a little bit, like read the first two parts of an answer and then just pick it? Because if an answer isn't 100% right, it's 100% wrong. So for this one, we're looking at the order of DNA synthesis. We need to unwind the DNA strands that will occur with DNA heligase. We need to pair our nucleotides and then join, which would be through the action of DNA ligase. So I gotta say, team, great work here, and I'm always up for a little bit of positive reinforcement, so to speak. So I like to give out little stars. So well done here, team. Well done. Let's do another practice question here. So we're going to get another poll up and running. I'm going to have you work on question number 2. Now, one thing I do want to highlight in this question stem is the word not. And you can highlight in the question stem on test day. You can actually use one of the Alt key shortcuts. So if you wanted to do that highlight, it would be Alt H. So go ahead and work through this one, and we'll talk through it together. All right, so it looks like we're a little bit split between some answer choices here. Split between A, B, and C. So we're looking for what is not a requirement for DNA replication. So let's take a look at our answer choices, and as we do, think about how DNA replication works. Now, of course, in order to replicate DNA, you need your nucleotides. You need nucleotides. Those are the dNTPs. These are your nucleotides. So you certainly need these guys to undergo DNA replication. So we can cross out A as a possibility. And on test day, remember, you can strike out answer choices with Alt S. Now, ATP, this one's kind of like the silent killer for this question. Keep in mind that ATP itself is a dNTP. The other thing to consider about ATP is that it is required by DNA helicase. It takes energy to break hydrogen bonds. Hydrogen bonds are pretty strong. So we need an energy source for DNA helicase. So powering DNA helicase. So that's needed. Oh, see, an answer choice is starting to change in this poll. I like it. So we can cross out answer choice B. Primer. Well, remember, before DNA polymerase can do its thing, it needs a free 3' hydroxyl. So RNA primase will come down and lay an RNA primer from which DNA polymerase will extend. So we do need that primer as well. So this is required for DNA polymerase. So at that point, we need all of these. So we got to go with answer choice D. So this is certainly a tricky question. But if you focus on, thinking about what you need before you focus too much on the answer choices, that certainly can help you. So let's take a look now at DNA repair. So I want you to tell me in the chat, what are some sources of damage where we need DNA repair mechanisms to fix them? I will say there's one that can't really be fixed easily, and for those of you who work in biochemistry labs, that would be if you worked with ethidium bromide, that actually will induce insertion mutations, which can't really be corrected very well. So that's why if you ever work with ethidium bromide, make sure you wear gloves and your lab coat. When I was doing my dissertation, that was really the only time I ever wore a lab coat, and I'm a virologist. So I'm seeing heat damage, UV light, absolutely, Levi. Definitely induces thymine dimers. That's why I'm very proud of this ghost complexion, because it means that I'm not actively giving myself cancer by tanning. So UV damage, like we mentioned, can lead to TT dimers. I also have allemiscence mutations. Mutations can occur during DNA replication. So what will happen is we have some sort of UV light leading to damage. X-ray can be a little bit more problematic, mainly because it's higher energy. So this can actually cause double-strand DNA breaks, which actually has a different repair mechanism, which you don't need to know. But that's what can lead to transpositions and such in the DNA. So we have our damage. And whenever we have this dimer, notice we get this little bump. This is the same type of bump we would get, or at least similar as if we had a mismatch. So there's always going to be some sort of distortion in the helix that can be recognized. And the first step is to cut out that piece of DNA using an endonuclease. So we see endonucleases in, of course, in our nucleus. We also have exonucleases, which are often found in the cytoplasm. What does an exonuclease do? This is something that will become important when we talk about transcription and post-transcriptional processing. But I do want to kind of set that, kind of foreshadow it a little bit here. What does an exonuclease do? One thing I do like to look at are word roots. So if you look at endonuclease, endo is for within. So if I had an exonuclease, this is something that cuts outside. So endonucleases are cutting within the DNA. And exactly, Allie, the ends are what we see exonucleases do. I like to think of those as kind of like little Pac-Men that just eat their way in from the outside. And I see a lot of us talking about introns and intron removal. For introns and slicing out those introns, because we're cutting inside the RNA, that would actually be done by an endonuclease, since it's from within. Exo will always count from the outside. So if you are familiar with restriction enzymes, which we use in cloning a lot, if you've cloned stuff, I'm sorry, I know it's horrible. Those are all endonucleases as well. So our endonuclease is going to cut out this damaged region, and then DNA polymerase is going to come in and fill that gap, and then ligase will join the two pieces together. So there's 3 specific mechanisms, which all use this general process. It's just a little bit different in the recognition specifically. So we have base excision repair. I'll type these out for you. So base excision. This is where we remove just the base, and then other proteins will come in and recognize either an apurinic or apyrimidic site. Essentially is a naked site, it doesn't have a base. So then we cut out the region and then fill it in with new DNA. We also have nucleotide excision repair. This is what we use for thymine dimers, or if we have single strand breaks. So this is nucleotide excision. This is where we remove 2 to 30 bases or nucleotides, and then we'll fill it in. The other one is mismatch repair. And this is what occurs essentially during DNA replication. So as we're replicating our DNA, we do have other enzymes that are there to ensure that we are not making mistakes. DNA polymerase does have its own proofreading capabilities, so if it notices it put in the wrong thing, it can essentially do a little backspace key, cut out that nucleotide and add a new one, and that helps to increase the fidelity of our own DNA polymerase. Now, if we are doing mismatch repair, how do we know which base pair to remove? Because we have 2 strands. So if we had to just randomly guess, we'd have a 50-50 chance of either correcting the mutation or essentially ingraining it into our genome. So how can we tell the difference between the newly synthesized strand and the old strand of DNA? What distinguishes parent from daughter strand? This is also something that is used in gene regulation, which is often associated with silencing. You got it, Hannah, Miguel. It's DNA methylation. So essentially, we recognize the methylated strand as the parent, so we do the excision on the not methylated strand. Absolutely. And this brings us to another practice question. So I'm going to have you work through question 3. Again, I want you to think about what is true of both DNA replication and DNA excision repair, where we cut out the base or the entire nucleotide as you work through this. So always make a prediction before you look at your answer choices. That will help to increase your accuracy. Go ahead and think about this one. Go ahead and pull in. All right, looks like we're split between 2 answer choices. We're split between answer choices A and D. So we know we need DNA polymerase either to synthesize the DNA in replication or to fill in the gaps with excision repair. We need DNA ligase for the lagging strand to join those Okazaki fragments in DNA replication. We also need it in DNA excision repair, since when we fill the gap, we do have that little nick that we need to fix. So we certainly need both of those enzymes, so we can cut that out. But then the question of the ages becomes, do we need an endonuclease? Well, remember that during DNA replication, we also are fixing mistakes. That mismatch repair occurs during DNA replication to ensure any mutations or oopses that DNA polymerase made is not going to be there forever. So we do need endonucleases for both DNA replication and excision repair. So here we would want to go with answer choice D. So nice work so far, team. That one was tricky, but this is the types of tricks that the MCAT loves to throw at us. And that's why it's so important to do practice questions, especially full-length practice tests, to really start building your pacing, your strategy, and also just to expose yourself to a lot of different questions. I say practice makes permanent. So the last thing we're going to look at is regulation of gene expression and, of course, transcription itself. Now, in doing this, we're really going to be focusing on prokaryotes in terms of control. In eukaryotes, we have enhancer elements, we have suppressor elements. There's a lot of different factors that control gene expression. And in fact, a lot of those elements can be located thousands of base pairs away from the promoter itself. So the promoters can be just really, really big. In prokaryotes, we don't have that kind of space. Not as complex. So with smaller genomes comes a more controlled specific unit. And that is what we define as the operon. So an operon itself will consist of a regulator. That's the gene for the regulator. What does the regulator encode? As you type in, I do want to note that the regulator gene doesn't have to be next to the promoter. It can be anywhere else in the genome, and that's usually the case. It's not usually that you have your regulator right next to your promoter element. It can be somewhere else on the genome. But our regulator is what encodes the repressor. The regulator encodes our repressor. And the repressor is what binds to an operator. So just to define our other sites, the promoter, this is where RNA polymerase is going to bind and do our transcription. We have the operator, and this is really the control point. We don't have all those fancy enhancers and suppressors and all those different proteins. We essentially have the repressor, which says, the operator, which says yes or no. Now, in the case of the lac operon, this is what we call a inducible system. An inducible is essentially turn it on. Now, I'm kind of curious here. Do you think we would have inducible systems for anabolic or catabolic processes? I think it's going to be anabolic, where we are building things, or catabolic, where we are breaking things down. It's going to be most likely to be inducible, where we can turn it on. Catabolic, absolutely, Miguel, Aaron. And let's think about why that would be. In catabolic processes, we are breaking something down. If we think back to our lac operon, if we don't have lactose, there should be no reason to make the enzymes we need to break it down. So our structural genes will consist of several genes, not just one. This is what we call polycistronic, where we have several genes that will be transcribed and then translated from the same messenger RNA. But without lactose, there's no need for the cell to expend the energy to make the proteins to make lactose. So the default of this system is off. Now, if we don't have glucose, because bacteria will always preferentially use glucose, if we have lactose hanging around, it now makes sense to make the proteins to break down lactose. So what happens is lactose acts as the inducer. It binds to the repressor and causes it to dislodge from the operator. So now we can make the genes. And transcribe, we can make our protein. So the goal of an inducible system is essentially to make the stuff we need to break something down when we have that thing present to break down. It's a way to ensure we're not wasting energy. Now, the other operon that we'll see is the trip operon. The trip operon is what we call a repressible system. So in a repressible system, this is very common for anabolic processes. So what we have is its default is on. The repressor itself is unable to bind to the operator. So in absence of our repressor, we're going to keep on making our structural genes. So here our default is on. So we're making stuff. Now, if we're looking at our trip operon, which is responsible for making tryptophan, it's certainly not going to make sense to continue making a whole boatload of tryptophan if we already have a sufficient quantity in our cells. So again, this is to really conserve energy. Now, our cells are very, frugal with their ATP, so there's no need burning it if we don't need to. So as we start to accumulate more tryptophan, it actually serves as the co-repressor. So now our repressor, armed with its co-repressor, can bind to our operator and shut down transcription. So what type of a feedback mechanism is this, where we have the end product serving to shut off the creation of more of that product? And this is actually how most of our biochemical pathways and honestly endocrine pathways operate. Yeah, Levi, Ali, Dominica, Alex, May, you got it. This is negative feedback. The product inhibits the creation of more product. There are very few positive feedback systems in the body. One of the most commonly tested ones on the MCAT is oxytocin, which stimulates uterine contractions for labor, which honestly, that one really makes sense as being positive feedback, because if it were negative, labor would never end. And then nobody would want to have babies, and then there would be no more humans. So absolutely fantastic work so far. Now let's take a look at transcription itself, and we're going to look at some of the specific elements in prokaryotes and eukaryotes. So promoter elements themselves are common in pretty much everything. We see these, we need it in order to make RNA, but we have different promoter elements. The Tata box is specifically found in eukaryotic genomes. The other really cool thing about this is for DNA viruses that replicate in our cells, like herpes simplex virus replicates in the nucleus, almost every single promoter of HSV contains a Tata box. Like there's more data boxes per gene in HSV than there is in our own cells. So viruses actually do take advantage of our own biology for their own benefit, which I think is pretty cool. Now, the sigma factor is a transcription factor that is in prokaryotes. And the rest of it, you know, just general transcription factors, RNA polymerase, and just our ribonucleotides are all needed in order to just start this whole process. Now, for elongation, of course, we need RNA polymerase. It's making our RNA. We have ribonucleotides. We also need helicase. Do you think we're going to need helicase in prokaryotes or in eukaryotes? Thinking back to just the other stuff that's on our genomes. This is one of the main differences between eukaryotes and prokaryotes. Eukaryotes actually have all of our DNA wrapped around in histones, which can be modified to make them either tight or loose on the DNA. So we do need helicase in eukaryotes, absolutely, as I'm seeing, because there's just more stuff to move. It's harder to move past. So that's something we need in eukaryotes. Oh, and Alex, HIV is TAT as well. That's really cool. I did all my studies on herpes, so I can tell you like more than you'd ever want to know. But about herpes, but I'm gonna hold back because that's not really gonna be useful on your MCAT. The last one we have are termination factors. We have hairpins, which are used in usually both. I will say that transcription termination is much more complicated in eukaryotes than it is in prokaryotes. It's still actually an area of active study in eukaryotes, so it's not likely to come up on test day because the mechanism is still kind of being elucidated. But we do see the Rho factor as our termination factor in prokaryotes. Our termination factor in prokaryotic cells. So now let's make some RNA. So we're going to look at our coding sequence here. Let's take a look. So here we're looking at just the DNA itself. So we have our promoter element. This is what recruits RNA polymerase. We have TATA boxes. This is one of the most commonly studied promoter elements, although most of our genes actually don't have them, which I always thought was really weird when I learned that. But it is the most commonly studied, so good one to know for testing. Cat box is just another example promoter element. It's not one that really comes up, so I would really just place your emphasis on the TATA box. Now, everything is measured. You see that -70, -25, that's all measured relative to what we call the transcription start site, and that's where RNA polymerase actually begins adding nucleotides. So it'll go ahead and start making our DNA, or sorry, making our RNA, and RNA polymerase 2 is the one that's responsible for transcription. We have polyadenylation signals, we have a cleavage signal, which also works with this GU-rich signal. But RNA polymerase 2 in eukaryotic cells will actually just like transcribe thousands of more base pairs than is needed, but then we cleave that signal and then just polyadenylate our final transcript. Now, what we get hot off of RNA polymerase 2 is heteronuclear RNA. It's not mature messenger RNA. So I want you to tell me below in that chat, what do we need to do? If we are a eukaryote, this does not happen in prokaryotes. What do we need in order to achieve a mature messenger RNA that we are ready to send out into the nucleus for translation. What types of modifications do we need to make? Absolutely, Diamond Alley, we need to splice, cut out the introns. Yeah, we need to process. And medicine, we have the 5' cap, poly A tail, the tails, absolutely. We got them all down here. Let's talk about the importance of each of these. Now, we don't need these in prokaryotes because prokaryotes don't have introns. It's all coding. And also, they, as soon as, this is actually really interesting in prokaryotes, as soon as you start transcribing, like translation starts. There's stuff that's like actively being transcribed, and then the ribosome hops on and it starts making it before like RNA polymerase is even done. So our bacteria are very impatient when it comes to protein synthesis. So we need to have that 5' cap. This helps with translation initiation. If you don't have a cap, it will not be translated. It's also for protection. And this is another thing that viruses take advantage of, specifically influenza, because it is an RNA virus, although it does replicate in the nucleus. But it actually steals caps from our messages and adds it to its messages to translate its proteins. So influenza is a thief. It is the cap snatcher to take advantage of our own translational machinery, which I've always thought is really cool. We have the poly A tail. This is for protection. Again, protection against that RNA, essentially RNases, those exonucleases in the cytoplasm. So our exonucleases, I always like to think if I had a messenger RNA, I have my poly A tail. These guys are like Pac-Mans, and they just chew in from the edges. So this would be an exonuclease. So Pac-Man chewing in from the edges. So you can actually think about the poly A tails kind of like a fuse or like a timer. So the longer that poly A tail, the longer that messenger RNA will be available for translation. The last we have is splicing. Always remember, introns go into the garbage and exons are expressed. So introns into the garbage, exons expressed. So we have 2 more questions to look at to reinforce what we're learning. So I'm going to have you work on number 4, and we'll come and review it, and we're really going to focus on how, in a question like this, where our answer choices are long, you can use their structure to help answer questions. All right, so go ahead and pull in. All right, so liking where we're leaning in this poll. So we're looking at specifically eukaryotic mRNA production. And notice we have some pretty nice parallel structure. We have where it's transcribed, followed by where it is processed. This is what we call, we call this a 2x2. This is one of my favorite strategies that I learned as a student. So we really only have to think about 2 things to answer this question. Where is it transcribed and where is that RNA processed? Now, if we think about it, transcription, of course, has to be in the nucleus. So we can start eliminating stuff here. It's not in the cytoplasm. It's not on the ribosome. It's not in the cytoplasm. So we've eliminated 3 answer choices. And if we just want to double, check yourself before you wreck yourself, post-transcriptional processing is also in the nucleus. So we'd want to go with answer choice B. So I just say that's where we were leading in this poll. So well done here, team. And again, use those answer choices to help you. And that's why you want to make sure you read those answer choices really carefully. So nice work, team. We have one final question to look at. Let's take a look at number 5. This is another one you want to be careful with because there is a trick to this one. We'll give you some time to work on it. Go ahead and pull in when you have your answer. Okay, go ahead and pull in if you haven't already. And let's think about some elimination that we can do here. So this question specifically asks for the RNA strand. So RNA should have uracil in place of thymine. So we can start eliminating some answer choices. Now, here we can get rid of really only one answer choice, and that's answer choice D. So already down to at least 1 in 3 chance. Now, the next thing we have to consider is the strand directionality. So if we look at our strand here, we have G, A, T, T, C, A, G, T, A, G, T, C. So this is our DNA. Our RNA, remember we have to go from 5' to 3' to make it, would then be the anti-parallel strand. So we have GAC. I know those are going to be my first 3 nucleotides, going 5' to 3'. And if I look at my answer choices, my remaining answer choices are all different in those first 3 nucleotides. So I don't actually have to transcribe this entire thing. We just need to transcribe just enough to get to an answer. And that's the idea of working smart, not working hard on test day. Don't do more work than you need to do. I always tell my students to be strategically lazy. And that might be the only time in your life that you'll have a teacher to tell you to be lazy. Don't do more work than you need to do. So at this point, it's got to be answer choice B. And the real key to this question is to remember the directionality in the strands. That was the trick built into this one. And if you fell for that trick, don't feel bad. A lot of people do. But again, that's another opportunity to learn from your mistakes. Every mistake you make is a learning opportunity. So I do want to say absolutely fantastic work here, team. I know that this is, maybe some information you haven't seen in a little while. That's one of the things I personally love about MCAT channel is you can really dig into some of the more high yield science and, get more, fun times. So I'm going to turn it back over to Kristen to talk to you about some other stuff, how to prep, and hope to see you around sometime on channel. Great work, team.
All right, everyone. So what did you think? Justine's pretty awesome. That's what I know. But what did you guys think of the lesson? Did you learn something? Yeah, I agree with Deborah. She did an awesome job. I always love watching Justine teach. So it's really cool to have channel opportunities in our free events. We absolutely love having them. So I kind of wanted to frame for you what a channel session is like. And I see that lots of you guys loved it. Crystal says loved it. She is cool. Pretty awesome for sure. So MCAT Channel is a cool resource that comes with our comprehensive classes. Basically, it is not our core class sessions, but an extra. So MCAT Channel meets 6 days a week, Monday through Thursday, Saturday, and Sunday. And it is 1 hour sessions each. Justine went a little bit over an hour because we had a little bit of extra time tonight, but we do normally keep them like right at an hour, and each of the topics is in an hour session. Sometimes a topic might take more than an hour, so we'll have more than one session associated. So one of my personal favorite channel sessions is thermodynamics, which I feel like is another one of those topics that students tend to need some help with. So thermo is pretty fun. We have 2 different sessions on that. One of them is kind of teaching more about energy transfer, and then the second session is more like applied thermodynamics with practice. So those are just a couple of examples of what channel could be. You know, we also have things like CARS strategy practice, you know, kidney sessions, enzymes, the list goes on and on. So channel's a very awesome resource. There are 10 teachers total that teach on channel, Justine and I included, and then we have some other awesome teachers as well. So it's always fun watching people teach a channel session. So how this fits in with our course in general, just to kind of orient you guys, because I know some of you have seen some different sessions this week. Channel is something that we would recommend to you at different points in the class based on what the topic is. So this is going to be an important thing because a lot of students ask me, how do you recommend getting through all of the content? So what we will do for our students is tell them, okay, you need to read biology chapters 1 through 3, general chemistry chapters 2 and 3, and you would get ready for class. Before class or after class, we might also say, hey, you should really attend the thermodynamics session. That would be a good one to go along with this material. And it's often that we will also talk about one of those topics in our core class sessions. So our core class sessions are 3 hours long, and they are at a set time if you're an in-person or a live online student. In the 3-hour core class sessions, we do some content. We call it high-yield science content, and we have kind of these interesting little prompts. They're almost like mini MCAT passages, and we will talk about content from the perspective of one of these mini passages. So that's kind of how a core class starts. Then we'll go into more of the strategy piece of class where we'll talk to you about, okay, how are we going to approach questions? How are we going to approach passages? As we go through those, of course, we cover content as well, since content is necessary in order to answer questions and understand passages. So yeah, MCAT channel is pretty awesome, Emma. We usually have 2 to 3 sessions like all year round, and then in the summer, oftentimes it goes more than that. Sometimes we'll have like 5 sessions in the summer because that tends to be some of our busiest time for people who are prepping. So normally it's in the evenings, Monday through Thursday, middle of the day, Saturday, evening, Sunday, and then we kind of increase that in the summer months. So we'll have some afternoon sessions like 3 and 4 p.m. Eastern. So it is a fun resource. I think it fits in really, really nicely with our core class sessions, and I'll talk to you guys a little bit more about, you know, if you aren't really sure how you're going to prep yet, we can talk very generically about what are the things you need to do in terms of prep that like you might not have thought about, and then we'll talk about how you might be able to utilize Kaplan to fill in some of those blanks. So if you haven't already, I do want to remind you guys that there is a drawing tonight, and we'll be announcing that at the end. So if you have not already, you should definitely register for a chance to win. You can get 6 months of Help Suite, which has a lot of our really awesome content-heavy lessons. So you can find the link right above the chat. So I highly recommend that you take a look. and you register for a chance to win. Okay, so I'm going to head us back over to where we started, and we're going to spend a little bit of time just kind of working on a list of the things that, yep, for sure, you'll want to do that during your prep journey. Okay, so I kind of want to source the information from you and see what you guys have already thought about. So if you have an idea of what you might need to think about, tell me in the chat, what are some things that you already realize are going to be pretty important when it comes to prepping? We're just going to make a list of that kind of stuff. So one thing that I feel like is probably pretty obvious is content. When it comes to content, though, I've had students ask me before, is it a good idea to go back to my undergrad textbooks, which I still have some of my undergrad textbooks, and I graduated from college 10 years ago this year, but I still have some sitting around. You may have yours handy from some of your prereq classes, but I do not recommend using undergrad textbooks or even undergrad notes in terms of studying, even for that type of material. The reason I say that is it's way too detailed. You do not need the same level of detail that you did, for example, when you took like organic chemistry 1 and 2. So it's important that what you're doing is MCAT specific content. Now, Bex and Valentina have another super great recommendation down here, which is making a schedule. Now let's talk about making a schedule in reference to the content part, and then I'll talk about how to fit in the practice part. So some of you guys might already have the Kaplan review books, and those books are split up so that there's one book for each of the main science topics. Now I don't recommend doing like the entire biology book and then moving on and doing the entire Gen Chem book and then going, okay, I'm gonna do the whole physics book. It's not really a good approach because you won't have seen some of that material, like the entire subject, in a really long time once you're to the end. So I think it's a great idea to kind of couple chapters of one, then switch topics, couple chapters of another, then switch again. And the end of chapter quizzes are a good resource to determine if you have that material down. Another thing I see students do a lot is, initially spend like 3 or 4 hours on a specific chapter, really trying to memorize every single detail and get it down. Now, I understand why there's a temptation to do that, because if you're pretty rusty on the material, you might feel like you need it. What I would really urge you to do, though, is consider a first pass, and then second pass, third pass, and whatever's needed when you go through the material. So I'm going to even put that up here. So what I mean by that is, when you're first looking at it, it is just a review. You don't need to know every single thing and memorize every single detail. So approaching it from a, hey, I'm going to review this the first time, see if it sounds familiar, just kind of get accustomed to it again. That is a good way to handle that content the first time. Because if you spend 4 hours on every single chapter, you're really going to take a long time to ever get to practice and to even get through all the material. So try and keep your first hour, hour and a half to each chapter or specific topic of one of your major sciences. Keep it shorter. Hour, hour and a half is a good amount of time for you to read and review, but you're not taking notes, you're not memorizing things. Now you might say, well, don't I need to come back and memorize those details and make sure I've got it down? Yes, but you likely won't have to do the same level of review for every single chapter. So keep a running list. Keep a list of the things, okay, I need to go do a second pass of this and this and this. And then if you do it and you're like, oh man, this is still some of the hardest material, I'm going to have to come back to this again. Then you might need a third pass. But some of the material, you'll be okay getting away with a first pass. Now if you're thinking, well, what if I thought I knew it and I don't later? That's okay, because when you get to the practice items, like some of you guys are mentioning the full-length practice tests, you guys will get to the point where you'll see what you're missing. So if on a full length you're like, yikes, I thought I understood replication and transcription pretty well after Justine taught it to me, but I missed some questions. Then it's okay. You can go back and do another pass of that material. So that's kind of a general idea of how to approach the content. Now, practice is another huge portion of this. Full-length tests are a part of it, but are full-length tests all you need? Are they all you need? Some students do use them as the main practice, but I do not think that's enough, like not by a long shot. We recommend taking 8 to 10 full-length practice tests for an ideal scenario, but even that is not enough practice because you're not doing practice as you're reviewing the material. Ideal would be, okay, I'm going to review the content topic of DNA replication and transcription. Then I want to do some passages and some questions to make sure I've got it down. So all of that kind of practice is super important. So I'm going to add practice questions and passages to our list. Another thing that's super important that often gets forgotten is reviewing your practice. If you make mistakes, but you don't know what those mistakes are, and you don't know what you need to do differently next time, it's unlikely that you're going to see major gains. So it's super important that you review every full length you take. It's important that you review every passage you do to make sure you understand the things that you missed and what you're doing well on. What else do you think we should add to our list? What else do you think we should add? I haven't seen anyone mention strategy, but if you guys have attended any of our other sessions this week, you may realize that strategy is a big part of this test. And I'll even throw into that strategy and test expertise. Knowing how the test maker is going to ask the questions, knowing the types of traps they're going to have. All of those things are super important when you're going through and learning the best approach to take on the questions. All right, what else? What else should we put on the list? Any other ideas? Do any of you guys have a approach that you use when it comes to memorizing discrete facts? You know, maybe you would want to look at something like flashcards, or if you do kind of like a quick equation sheet. You know, those are kind of things that really can help you as you're moving forward. What I've done with equations, because there are a lot that you have to memorize, what I did when I first started studying for the MCAT, I went through the Kaplan review books that I had back then, and I would write down every equation I came across. And then once a week, I would go through and rewrite that list. And then I would try and eventually write it without looking at the previous list. Then I would start trying to group the equations with similar variables, the equations that I might have to use with one another. And once you can do that, then you've pretty well got them memorized. But memorization is not the only part of it. You also have to know when to use the different equations. So practice is critical. Another huge mistake I see students make is not doing practice early enough. A lot of students feel like, well, you know, I need to make sure I have all the content down before I start doing practice. If you wait that long, it is way too late. So what I think you need to do when it comes to a good schedule, what I recommend is in the first half of your studies, you do about 2/3 content review and 1/3 practice. Then when you're in the second half of your studying, you're going to shift towards more of a 2/3 practice and 1/3 content. And at that point, the content you're doing is based on the things you're missing when you're doing that practice. Okay, what else? Can you guys think of anything else that might be helpful? Anything else that you guys think would be a useful approach? Okay, well, let me just ask some things that these are not something that every single person has to do, but there are some things that you got to ask yourself to figure out what is going to be the best approach for you. So for instance, how many of you guys feel like you can learn content well from a book? Like textbook reading. I bet some of you are totally fine with that, and maybe you did that for a lot of undergrad. And, you know, I had some classmates in college that like really didn't go to class because they were fine just reading the book on their own. Okay, well, that's one approach, and if that works for you, that's perfect, then you do that. Now, I bet some of you also realize that just reading from a textbook is not the right approach for you, and so you need to figure out what you are going to use. We have different resources because different students are used to and excel with totally different things. So we have these short things called science content review videos that are 5 to 20-minute videos that go over a very specific topic. Then we have things like MCAT channel that go over a topic in a lot more detail for an hour-long session. And then sometimes those hour-long sessions stack up to even cover more, like in the example of thermodynamics. Then we have our regular class sessions, which are 3 hours long. And they cover practice, they cover strategy, they cover content. So having that instruction where you can interact back and forth like you could with Justine in this lesson is really important for some students. Now, some of you guys might also be like, well, when I had larger classes as an undergrad, it didn't really help me as much as I needed, and I did better with a tutor. Those are options as well. These are all things that you guys should think about early on so you are making sure you're studying the way that is going to work best for you. Christy asks, how can you focus on practice based on content you know? So I do think it's a good idea to practice the things that you're feeling good about so that you can really make sure that you know them as well as you think you know them. So we have a lot of resources that are broken down based on the chapter. So our passage-based questions are just like that. So for instance, if you studied a chapter of biochemistry, then you could go do the PBQs, which is what we call them, passage-based questions. You have 3 or 4 passages based on that chapter that you're going to be able to really kind of assess your knowledge and assess your mastery of that topic. So there's lots of different things, and I think depending on what has worked for you in the past, you could use a similar strategy here. One thing that I think that would be a mistake is for you to put it off really close to your test day. I mean, I feel like we all kind of know that, but it's one of those things that it's easy to let life get in the way. Do any of you guys feel like it's going to be really hard for you to fit in studying for the MCAT? Do any of you guys feel like you have competing priorities? Maybe you have classes, finals, a job, volunteering, shadowing, research, other extracurriculars. It's going to be super challenging to fit this in. So what you need to do is, like we said, make a schedule. You literally need to go week by week, understand what you need to do that week, understand how you're going to prioritize it against all of your other activities. These are the things that are going to help you actually accomplish them. I'm a horrible procrastinator, and I can tell you that it's freshly on my mind from when I waited till the Sunday before my taxes were due to even start downloading TurboTax software. So procrastination has been a challenge for me in college, in grad school, and still to this day. But I think having a good schedule in place is one of the things that you can do to really help yourself. Now, if you have other tricks that work for you, maybe having a study buddy, accountability partner, maybe knowing that, okay, I have to go to class because that's going to keep me motivated. Any of those things that have worked for you in the past, it really comes down to know yourself, know what works for you, and do that. So I'm going to now go over with you guys some of the different class options that we have available. And so once we talk through that, I will kind of just open it up to any and all questions that you guys have. So let me Mary, this one I cannot get to move to any of the hyperlinks. So if you could try and figure that out for me. Oh, maybe I got it now. Give us 1 second as we try and figure out what's going on with our PowerPoint. Okay, hold on 1 second. Nope, still not working. Okay, so I'm just going to walk you through them. Our little way of kind of showing you in a table isn't going to work tonight, but I will walk you through the different resources and explain them. So we have 3 different tiers of prep. We have our MCAT prep, which is comprehensive. It includes 11 Kaplan practice tests. It includes all of the AAMC materials, including the 4 AAMC practice tests. We have the science content review videos, the review books, the passage-based questions, quiz bank, section tests. The list goes on and on. Then we have MCAT Prep Plus. Prep Plus is kind of cool because it comes with one-on-one coaching in addition to your class. So you're going to meet with your MCAT coach on 3 different occasions, at the very beginning, towards the middle of your prep, and at the very end. So you would kind of meet with that coach and figure out a good game plan together. So that's super helpful. Okay, it looks like we have got one working now. So let me go ahead and bring this on. There we go. Now you guys can follow along with what I'm talking about. Okay, so the first column right here is the one I was just mentioning, MCAT Prep, and then this is Prep Plus. So one-on-one coaching is just one of the things that comes with Prep Plus. Now, in addition to that, we also have the biochemistry foundations and the behavioral sciences foundations. This is perfect for those of you who haven't had biochem or won't have biochem, and then same thing with psychology and sociology. So that is Prep Plus. Then we also have private tutoring. So the private tutoring is just what it sounds like. You'd be meeting one-on-one with the tutor. This package does come with a class. It comes with all of the things Prep and Prep Plus come with, plus your one-on-one tutoring sessions. And there are different hour package levels based on what you think you need. Now, in addition to the different tiers of prep, we also have different formats of prep. So one of the formats we have is called Live Online. It is kind of similar to what we've done tonight. It's interactive with chats. You can ask questions, you can poll in, but you can also come to class from the comfort of your own home, or if you're on travel, you can just bring your computer with you. We also have in-person. So if you feel like maybe you get slightly distracted in this setting, or you just need the, more like, okay, I'm going to be in class, so I have to pay attention because the teacher can see me. Some people just prefer that a little bit better so that you can actually be in the same room with your peers and the whole thing. Then we also have self-paced. Self-paced is just what it sounds like. It is work at your own pace. If you want to watch class at 3 o'clock in the morning, you can do that. In-person and Live Online are at set scheduled times, but self-paced, you can watch it whenever you want. It's pre-recorded videos. You're going to go in, you're going to log into your CapTest account, and you're going to be able to watch that video whenever it's convenient for you. So again, lots of different options for lots of different students that have completely different needs. So there's not just one program that's perfect for everyone. So I think it is important that you guys do some reflection and figure out what is going to be the best option for you. Now, I also want to give you guys some discounts that you can use for tonight. So the first one I'll mention is if you are an AMSA member, American Medical Student Association. If you're an AMSA member, you can get 10% off. So you would use your 6-digit national member number, and that's how you can sign up. You would call 1-800-CAP-TEST to utilize that discount. Now, we also have a military discount. It is 15% off. So this can be if you are military, if your parents, your spouse, siblings, any of those things, you can get 15% off. And that one, again, you're going to call 1-800-CAP-TEST to enroll using the military discount. We have another discount, which is just for saying thank you for attending tonight. So you can use the code MKGL181144. I'll say it again, MKGL181144. And that will get you $200 off any of the class options. Just pick the one that you would like. Then we have one final discount I want to mention. This one is our biggest discount. It can get you up to 60% off of the class. And it's called Starting Line. And it is a tuition assistance program. It is a pretty amazing program. It is a need-based, so we give lots of scholarships to be able to afford classes. So there are a couple of requirements. You have to be a current student, you have to have a GPA of 3.0 or higher, and you have to have, the need for it. And the way we figure out the need for it is using FAFSA, since it's already a need-based financial aid system. They have something called EFC score, expected family contribution. If your EFC score is below 4,000, then you will be eligible for our starting line program, and you can get anywhere from 25 to 60% off. Now, none of those discounts are going to be stackable, but you should certainly pick the one that's going to get you the most money off. Okay, so Anika says, I noticed in person most of the class is shy or reluctant to talk, and for live online, everyone is super involved. I do think that is pretty true. I think sometimes in class it is, who wants to raise their hand and be like, well, I don't get it, when everyone else seems like they understand. But usually once one person says, I don't get it, then more people are willing to, or more people are willing to ask questions or participate. And I find that that does happen more with live online, generally more students, so more people participate. Participation feeds participation. So I absolutely love live online. It is my preference to teach as well. And it's pretty nice being able to log in from the comfort of home. Okay, Emma says, is there an actual difference between Self-Paced Plus and Live Online Regular? Yes, there is, Emma. So Self-Paced Plus is going to be core classes that are still in that self-paced format where you log in and you watch a pre-recorded video. The plus part of it gets you the foundations of biochem and the foundations of behavioral sciences, and you get a coach. Whereas Live Online Regular, you're going to have a live online class that is live. So it's like you come in at a set time, you meet with your teachers. It would be just like this. You have teachers on camera, teachers off camera, set time. But then you wouldn't have the coaching part of it. So, I think both of those are good options, just depending on what you need. Again, I absolutely love live online, but I realize it doesn't work for everyone. My husband, for example, if he were studying for the MCAT, he actually works for the FBI and isn't interested in the MCAT, but he does have a schedule that rotates every 8 weeks. So, a set time would probably not work for someone who has a schedule that changes like that quite a bit. So there are certainly advantages to each of them. Crystal, great question. So the live online option will allow you to view class later because what you're going to have access to is the same self-paced videos. So you can log in at any time if you miss class and watch the pre-recorded video, or if you miss class but want to attend another live class, you can attend any of our ongoing classes. We have a bunch going on at the same time, and you can attend any of those as makeup sessions. Great questions. So if you guys have other questions, please let us know and we would be happy to help. I will go ahead and show you guys some of our upcoming classes just for some examples. So it looks like Mary recently started one. Mary's with us in here tonight, but you can just get a sense. Some of these have already begun. We have one that is starting in a couple of days with Jim. These are all the April classes. So I'll also show you May. You can kind of see, okay, some of these are once a week in the evening. We have some that are twice a week on the weekends only. We have some that are 3 times a week during the day. Just tons and tons of different options. I have a class that is starting on May 3rd, Thursdays at 6:30. You will see Justine and I for sure if you end up in a class because you'll come see us on MCAT channel, which we would be super excited about. If you want to see the full list of classes and all of the different schedules associated with them, you can go to captest.com/enroll/MCAT, and you can see the full list of live online classes, and you can also search there to see if in-person classes are available in your area. Now, one last thing I want to mention to you guys is, it's a little scary to spend a decent amount of money, which these classes are not cheap. It's a little scary to spend that much money on a class and be concerned that you're not going to improve as much as you want. You're not going to get the MCAT score you want. So one of my favorite things we have is a higher score guarantee. This guarantee allows you to repeat the class. So if you take the class and you do not see the improvement you want, you come back and you repeat the class. Ultimately, we want you guys to get the score that you want to get. That's the whole point. That's why we do what we do. So you get really 2 classes for the price of 1. If at the end of your first one, you're like, I'm not ready to take the test, come back and repeat. If at the end of the first one you're like, I think I'm ready to take the test, I'm going to take it and see how it goes, then you get your score back and you're like, not quite where I want it to be, then you can come back and take the class again for free. So it's a really nice kind of little insurance policy you've got just in case you don't see the improvement you want the first time. You can come back and do it all again. So if you have other questions, let us know. But that is everything I wanted to share with you guys. And the last thing I will do is share with you the winner of our Help Suite 6 months for entering the drawing tonight. It is Anya Suros. So Anya Suros, congratulations. It's a pretty awesome little prize for attending tonight. So thank you guys all for entering. If you do have any other questions, please, again, let us know. We hope that you guys will come back and join us for some more of our free events. We have lots of them: practice tests, sample classes, boot camp, MCAT channel sample. There are some good ones. Christy, it takes 30 days about to get your scores back. If you go to the AAMC website, you can look based on the test date and it will tell you exactly the date you can expect your score back. So thank you guys so much for joining us tonight. Thank you to Justine for an awesome lesson. I love watching her teach, and all of our other teachers who have been behind the scenes helping out. It's been great to work with all of you guys. Again, we really hope to see you in one of our upcoming events or one of our classes or channel sessions. And I wish you guys all the very best of luck with your prep moving forward. I know you guys can do it. Just stick to it. It takes some blood, sweat, and tears, but you have very admirable goals. You're going to be great doctors. So I can't wait to see some success from all of you. I hope you guys have a great night, and we hope to see you again soon.